And we did a very rough estimate of how much of the stream bed was actually changed by lamprey. So overall, about 2% of the wetted area of the stream bottom was actually affected, but if we took out all the parts that weren't spawning habitat, the deep, slow, muddy pools, and we focused just on riffly, rocky habitats that Atlantic salmon will probably use as well, about a third of the stream habitat was actually impacted by lamprey, a relatively small population. So they have the capability of exerting a large effect in large numbers, probably. Pretty good diversity of aquatic insects in their nests. Most of them were these chironomids, these midge larvae, and also net spinning caddisflies, and then some mayflies. And it turns out that insects living on the stream bottom here, the benthos, much higher densities of these insects, especially in the mounds, compared to an undisturbed reach. Probably made very good habitat for these, especially these little caddisflies, to get in the nooks and crannies and build their catch nets. And then drifting insects. We compared drifting insects that came off of an active nest versus just a spot that lampreys weren't disturbing, and it turns out there was a much greater abundance, biomass, and diversity of insects that were drifting when lampreys were dislodging them through their spawning activities. That's not a surprise. And so all of those taken together, we would say that the spawning behaviors increased the habitat complexity, it increased insect abundance, and it coarsened the substrate. And so that would probably have some pretty impactful benefits to drift feeding fish like Atlantic salmon that feed on drifting aquatic insects, or even adult Atlantic salmon that would spawn over similar habitats. And let's see. I really like, there we go. I really like feeding of trout and salmon for whatever reason. I like to fly fish. I think the behavior of fish feeding is fantastic. So started looking at the impacts of sea lamprey spawning on how fish actually behave and feed. So there's a couple lamprey down there that are actively spawning, and you can see there's just minnows stacked up behind these things. There are at least 3 species of minnows in there. There's a spawning right there, and you can see these big common shiners are popping right into that plume, and they're feeding on presumably eggs or benthic invertebrates that are dislodged. But these minnows follow these schools of lamprey around, and very, very aggressively darting in and out, feeding on the disturbance from the lamprey. And I could watch those all day, but I won't subject you to that. So then what we decided to do was do some snorkeling and some videography to looking at the foraging behavior and the energetic costs and benefits of those small drift feeding fishes that were associated with lamprey nests. And let's see, maybe this will work. So put up some observation grids and go back, and you can measure depth and velocity later, and you can look at individual fish position against the grid and figure out how fast they were swimming. You can count their tail beat frequencies. So you can see they're darting in and out of those plumes. And they're swimming. This is very, very fast. Probably a huge energy expenditure for these fish. They typically don't swim that fast, but they're in fast currents here, maintaining their position. And so after about 100 hours of videos like that, I haven't done a whole lot of analysis, but from some pilot data, some preliminary thoughts that I had was these minnows respond very quickly to spawning activity, and they engage in what otherwise would be risky behavior. They're out in shallow water. They're in a whole aggregation of very large fish that are thrashing around. They're vulnerable to predation there. They form these really dense roving aggregations and follow these spawning lamprey around. They occupy really high velocities, and they miss a lot of food. They don't capture a whole lot. So the fact that they keep on doing this over and over and over again suggests that at least sometimes there's a huge energy benefit associated with feeding behind lamprey. We cut open some of their stomachs, and sort of disappointing from my aspect, and probably from the fish's aspect too, is a lot of times their stomachs were filled with sand and silt and not insects or eggs. So I think the minnows that get there first are the ones who get the smorgasbord of eggs and insects. The one who get there late, all they get are silt. But the next step would be to test this with juvenile salmon. So then the last part of the research I want to tell you about, I've got a PhD student who just finished up. He's actually gainfully employed now as a professor, so I guess I've produced a clone of myself to go on and doing meaningless stuff. Well, anyway, so he was very interested in looking at how lampreys serve as vectors of energy and nutrients and how they link stream habitats, forest habitats, and oceans. And so just a slightly different life cycle diagram here. So we've got these these larval lamprey that are filter and deposit feeding inside a stream in these slow, silty areas. And these streams are typically really heavily forested, and most of the food that comes into these lamprey probably are from leaf litter, little tiny pieces of leaf litter that are broken down, maybe some algal cells that are sloughed off. Well, anyway, so these guys transform, they go down to the ocean, they pick up huge reserves of marine-derived nutrients and energy. They do about 98% of their growing out in the open ocean where it's productive. They come back up, they spawn, they die, their carcasses decompose, and the question is, do the nutrients from the carcasses then end up into the ammocetes and improve their growth rate? And essentially these lamprey would be linking all these three different habitats. So there's some studies done from other places in a similar stream to Sajunkadunk. And so we use some of their estimates for how much energy and materials these lamprey deliver. And so just a back-of-the-envelope calculation suggested that in Sajunkadunk stream in 2012, about 300 lamprey would have brought something like 20 kilograms of nitrogen and a kilogram of phosphorus. So averaged over the entire stream, we're not quite sure if that's biologically meaningful or not. And so that prompted us to do a series of studies. So started with actually a previous grad student to Rob. We went through about 5 or 6 iterations of these types of studies. So we decided to do carcass experiments where we added carcasses, we put them in cages, and we put tiles downstream of them. And these tiles would collect any algae that would colonize. The idea that these rotting carcasses should increase production of algae in the streams. So we started by just dropping off individual carcasses in the stream. We did some chemical analysis, and we found that, first of all, for their body tissue, their nitrogen-phosphorus ratio was really high. It was about 20 to 1, and if you're familiar with a lot of aquatic ecology, about 6 to 1 is sort of the optimal phosphorus-nitrogen ratio. So these guys are very, very poor in phosphorus, probably because they don't have bones. But they're relatively rich in nitrogen, so if the stream is nitrogen limited, they could be a boon. If the stream is phosphorus limited, they probably don't do a whole lot. So very rich in nitrogen compared to phosphorus. And then we weighed these carcasses. I don't know if you've ever weighed rotting, soaking wet lamprey carcasses over time, but it's kind of difficult and it's disgusting too. But anyway, so they rotted pretty quickly. They lost about 60% of their body mass in the first week, and over 4 weeks, about 80 to 90% of their body mass under ambient temperatures. So they're decomposing really quickly. Early summer is also when stream temperatures are up and the metabolic needs of aquatic insects and fish are high. So this might be a really important pulse of nutrients and energy at a time when the stream's limiting. Well, then we decided to do this a little bit better, so we have a lab experiment where we took carcasses and we put them in these individual buckets, and we could measure the outflow of nutrients, and we could regulate the water temperature to get a better idea of how fast they liberate nutrients. And so up here you can see ammonium is at the top and phosphorus is at the bottom. And so we get a spike of ammonium and a spike of phosphorus really early on, especially at high temperatures. And the phosphorus is essentially gone really quickly. Very quick pulse and gone. The ammonium, it can be protracted out through the summer. So there's different nutrients that are coming in at different forms and different concentrations depending on temperature. So it's probably pretty complicated to predict what those nutrients will actually do for stream algae and invertebrates and whatnot. Then we went back to our carcass edition. Now we decided we're going to seed whole reaches with carcasses. So we tried doing high density, about 15 kilograms of rotting lamprey tissue per 100-meter reach, and low densities. We had upstream, downstream comparisons. And So that's what our streams end up looking like. There's, lamprey sort of scattered haphazardly around at some density, and you can see we're wearing our masks, and we rub the Vicks under our noses because the smell is just absolutely putrid. Absolutely, yeah, just terrible working with lamprey carcasses. And then we also tried to do some experiments on nutrient limitation. So we put in these substrates that have these little glass tiles on the top here, and they would collect algae. Algae would colonize. And then those vials were spiked with either nitrogen, phosphorus, nitrogen and phosphorus, or nothing as a control. So that would tell us which of those nutrients is the most limiting for the stream. And so we put these nutrient diffusing substrates above and below carcasses. We measured the algal biomass. We would measure nutrients in stream water. We'd measure some benthic invertebrate abundances, and then we'll go to stable isotopes a little later. And so for this experiment, we didn't see anything other than we found out these streams are nitrogen limited. So it didn't matter whether these tiles were just below a carcass or upstream of a carcass. There was no difference. The carcasses didn't do anything for the algal growth, but adding nitrogen did. So we were able to at least conclude the streams are nitrogen limited. And so keeping that in the back of our mind, lamprey are rich in nitrogen over phosphorus, so maybe lamprey could relieve nitrogen limitation. So then we decided we're just going to now just saturate the stream with carcasses. We're going to try doing anywhere from low to really high densities and see if there's a threshold or a cumulative effect that you need a certain quantity of lamprey in an area. Bomb? That is a carcass bomb, yes. There are probably 10 lamprey in there or so. Oh, yeah. And then we took out the Moab, the mother of all lamprey bombs, with these lobster cages with something like 50 kilograms of lamprey in them. Probably simulating an incredibly high spawning run, but maybe realistic. And it turns out that at least for algae growth, when you have a high enough carcass density, you get this cumulative effect where eventually algae respond. So these lamprey carcasses in high enough density will improve algae growth. So increase primary productivity of the stream. And then we wanted to see, okay, let's close the loop, see if we can connect the larval lamprey who are feeding on filter feeding detritus with the adult lamprey who have been parasitic, came back to the stream to die, to see if we can connect those two somehow. So the next step was to start trying to quantify the larvae, the amiseed abundance. And so here's Sajunkadunk Stream. Here's all the places where we found nests of lamprey. And we just broke the stream up into 6 areas, A through F, depending on what the substrate was like. And we used this, we just built a little sampler here and essentially dug out the substrate. And you can see the marks left there. So it's quantitative. We can get a known quantity of substrate. And so we moved an awful lot of sand and mud and gravel, for weeks at a time, just going out there, digging up the substrate, running everything through sieves. Oh, I guess you can't see it. The lighting's not that good, but you would see a whole bunch of squirming little amiseeds in there. And so we got some estimates of amiseed density. And so it turns out there were really only 2 hot spots in this stream. Everything else was relatively devoid of lamprey. But anywhere from 0 to 100 amiseeds per meter squared, which 0 is on the very low end. 100 is not quite high. There are densities of several hundreds in places like Great Lakes tributaries. On reaches, 8 to 30 per meter squared, so that gives us about 15,000 per couple 100 meter long reaches. So not huge abundances, but again, this is only a few years after dam removal, so these fish seem to be recolonizing and spawning pretty quickly. And then we modified our technique a little bit, and now we're using electrofishing to catch the amiseeds instead of digging them up. So we block off some sections near the shoreline. That's Tom Evans over there, who was a visiting grad student from SUNY ESF, who did some of his work in collaboration with us. So then we just did a survey around the Penobscot watershed, and in no particular order, these are some streams that we thought would have pretty high larval lamprey abundance. As many as 50 per meter squared. Again, not super high, but not inconsequential. And we found a whole lot of locations with no larvae. And so we have a pretty good idea of hot spots for lamprey in the watershed, and the idea is go back after the dams, the main stem dams and the Penobscot have been removed for a couple years, and see if those sites have responded. And then the question is, do larvae or other benthic animals, insects and mussels, actually assimilate the nutrients liberated from the carcasses? And so how many people here have heard of stable isotope analysis? A few people? So essentially, different forms of molecules like carbon and nitrogen and sulfur in some cases can give you clues as to the origin of the food source of an organism. Essentially, they are what they eat. And if you plot the relative enriching of the heavy isotope of carbon here and relative enrichment of nitrogen up here, essentially your primary producers are down here, stuff that eats them up here, and so on, and your top predators are up in this portion of the graph. So they're becoming more and more enriched with these stable isotopes. So what you do is you sample the tissue, you run a stable isotope analysis, and it tells you essentially where the feeding position of those organisms are. But another good thing about stable isotopes is it can also discern between freshwater and marine origin of food. So freshwater would be relatively depauperate in the heavy carbon and nitrogen. Marine environments would be relatively enriched with those forms. And so the adult sea lamprey would be coming into the stream bringing a really significant marine signature, very easy to detect. Presumably, the ammocetes would be feeding almost entirely on detritus, and they would be down in that left-hand corner. And we would expect if the carcasses, let's see, how do we go here? If the carcasses are feeding the ammocetes stable isotope signature would move up over time and become more like the adult sea lamprey, the carcass. And so we had a student who essentially did that for a bunch of organisms, but I'll show you a couple here. So predaceous stoneflies, here's the adult lamprey, the marine signature. Here's where the stoneflies started out at, and over time when they had access to the carcasses, their signature sort of moved towards the adult lamprey. Hellgrammites here kind of moved up towards the adult lamprey. So there's some evidence that nutrients from the sea lamprey carcasses are somehow getting into these predators. And then we have the juvenile lamprey, the larval lamprey here. Here's where they started out, and their signature shifted a bit over time. So there's some evidence that they're incorporating some of those carcass nutrients in their tissues. And then the last thing I'll show you, we're about an hour. We started a little bit late, almost done. So then my student, Dan, he built this simulation model. So he essentially said he built himself a virtual population of lamprey, and he said, okay, so these lamprey come into spawn, and some of the larvae survive, and they go through 1 year, 2 year, 3 years, so on and so forth, and how many years they spend in the stream is a function of their growth rate. Well, the growth rate is going to be influenced by how many nutrients that the adult lampreys bring in. And so essentially he's building a model here to link larval growth with nutrients brought in by the adults. And so he did a baseline run. You can ignore this sort of stuff at the beginning. The model is sort of burning in. But eventually when this model equilibrates, we see a difference between the number of adults that come back if they're allowed in the model to get nutrients from the previous spawners versus the number of adults that come back if they don't get to use nutrients from the previous spawners. So if they incorporate nutrients from the carcasses, populations can increase by about 20%. So not huge, but not inconsequential probably. But then the interesting thing was the nutrients from the adults stimulated growth of the larvae. The larvae have to reach somewhere between 120 and 160 millimeters to metamorphose and go down to be parasites in the open ocean. And so if they incorporate nutrients from the adults, they grow faster and they can metamorphose 1 to 2 years earlier. So they can leave earlier at a larger body size. So there's less mortality. They have a higher probability of surviving out in the open ocean. Again, these are simulation models. The next step would then to be ground truth this stuff. Well, that's essentially the story that I wanted to tell about sort of sea lamprey in general, but also some of the research that we've been doing in the Penobscot River. And I think there's a pretty compelling case to be made that these sea lamprey are probably very important components of this native fish community that used to be abundant here in Maine. Probably critical for the success of restoring Atlantic salmon because of their effects on the ecosystem engineering, delivery of marine-derived nutrients, habitat conditioning, and so on and so forth. So that's all that I have. And if anybody has any questions, comments, whatever, arguments, now's the time.
When they attach to a fish like a salmon or striped bass, do they detach after a certain point in time?
Yep, when they are satiated, they'll detach. And there was, back in the 70s, they built bioenergetics models of the lamprey that predicted how much blood a lamprey would take from a host fish and how long they would stay attached, and it was a function of lamprey size and water temperature. A large host fish with one or two small lampreys, it usually doesn't do anything to the host fish, but if you have a small weakened host fish with a lot of large lampreys, it can kill them. It takes something like 3 or 4 lamprey wounds to kill an average trout or salmon in the Great Lakes.
But not enough to cripple the survival of that other species that's being preyed upon.
Well, historically, no. Clearly, it doesn't do it in the ocean. There's just too many predator fish or host fish in the ocean. Lampreys are probably limited by habitat quality and spawning streams. Great Lakes are a different story because the native fish community was decimated by a bunch of different factors. Lake trout and Atlantic salmon went extinct or nearly extinct, the lake trout in Lake Ontario a long time ago. And that was Lake Superior lake trout, sure. So now there are exotic species, coho salmon, Chinook salmon, brown trout, steelhead. So left to their own devices, the lamprey population exploded and decimated those fisheries as well. So without the lamprey control measures, those exotic sport fish would probably be at very low levels. Of course, they're stocked as well. So it's an ecosystem that's already been heavily perturbed by a variety of reasons, and now exotic lamprey are coming in. It's a very different situation where lamprey are a native component and the ecosystem was relatively intact with native species that function like they used to. So very different story in the Great Lakes than here is the point. Yeah?
What are the main predators of sea lamprey, and how has their abundance kind of changed over time?
Not many predators. Certainly fish will eat them. People who fish for striped bass like using amicetes, little lamprey about yay big or so. Of course, they use eels too, so I would imagine anything a striped bass that looked like that, a striped bass would eat. Nobody really knows how important predation is for controlling populations or limiting populations of the larval lamprey. Probably not super important. It's probably more competition for space. When you crowd too many little lamprey in an area, their growth rate suffers. So predation probably doesn't limit them. As for adults, nobody knows. They're out in the open ocean. Certainly something eats them, but there are factors other than predators that are limiting lampreys.
See any interaction with eels in habitat?
Probably minimal. I would guess they don't interact very much. They just don't overlap directly in space and time very much, and they do very different things.
Do we have a sushi business potential with amicetes?
Man, if you want to eat them, go ahead, but...
Well, I think they are eaten in Europe, right?
Lamprey are a delicacy in Europe. And of course, there's the story that Henry VIII? One of the Henrys apparently got food poisoning from a batch of bad lampreys, and that's how he died. But yeah, lamprey were a delicacy and still are, but a lot of the species are endangered, so there's not a whole lot of commercial fishing for them anymore in Europe. Is it the Umatillas? One of the tribes up in the Pacific Northwest, the Pacific lamprey are very important for them for food and for cultural significance, and they love them.
Wow.
Are they very different fish? Or?
They're different species and different genera, but they're similar in the sense that they're both parasitic ocean-going lamprey. Yeah. My former grad student Rob Hogg, his son was part Umatilla or one of those tribes up there. So he spent a summer on one of the reservations, and he was catching Pacific lamprey when Rob was out here catching sea lamprey. And so he ate one of the Pacific lamprey, and he said they're not good at all. That was the report I got. But I wouldn't eat an adult lamprey, not after seeing what they look like coming up in the streams. Maybe out in the open ocean, but not when they're decomposing themselves.
They probably have to smoke them.
You could probably, I'm not sure if you get high off of them, but you could smoke them.
Oh, okay.
Ideal. So, go ahead.
What time of year do they migrate up into the streets?
So in southern Maine, here maybe a little more south throughout southern New England, probably April to early May. When you go up northern Maine, New Brunswick, maybe as late as June. And the ones that go very far upstream in a watershed will usually migrate sooner. The ones that only penetrate a small distance upstream will usually migrate later. So in Sajunkadunk, that's one of the latest runs in the state. For whatever reason, they're over by July 1st or so. But a stream next door, the Kunduskeg, they're done by mid-May. It's highly dependent on temperature and flow.
And how long are they before they die?
Less than weeks, days to weeks. Yep. They'll stage in large rivers below tributaries after ice out, but they don't do much milling around, and they don't start moving into the small streams until flow's gone down, temperature's gone up, and photoperiod is increased.
Nitrogen is often an issue when we get into talking about alewives and alewife restoration. Yep. And I wondered, if I heard you right, these guys are producing some nitrogen or inputting nitrogen
Yep.
into the system. So I'm going to guess it probably hasn't come up yet, but as we get into these issues around political implications of do alewives contribute nitrogen or take out nitrogen?
Yep.
You know, should we reintroduce or not? Sea lamprey might not fare so favorably when you've got a green lake to start with?
Well, their abundances are so low compared to alewife, which are, can be in the millions. Yeah. My guess is just by the sheer numbers, the alewife would be more important, even if most of them don't die, just through their excretion, their gametes. There was a study from some of the people at Yale. David Post works on alewife, and his students did a modeling study coupled with some data, and they showed that under certain conditions, the alewife could be net exporters of phosphorus. So they weren't concerned about nitrogen, they were concerned about phosphorus. So under certain conditions, they can export phosphorus, and it turns out that the phosphorus they bring in is actually on the same order of magnitude that a strong Pacific salmon run would bring. So alewife phosphorus, one might be concerned if the lake is already eutrophic. Maybe the first thing you might want to do is reduce the anthropogenic loading from septic systems and things like that and let the natural come in, but that's just my opinion. But I would say people probably wouldn't be concerned. There are other reasons people don't like lamprey beyond the fact that they bring some nitrogen up, but I wouldn't be worried about that.
So are there places sort of saying the numbers of sea lamprey are maybe inconsequential in terms of some of this?
Probably.
You did some heavy loading.
Yep.
When you were doing your experimenting.
Yeah.
Are there places where there are really high, loads of lamprey? Or?
I haven't visited places, but I've heard reports from the Sheepskit River that have tons of lamprey. There's a Pleasant River down east that there's some commercial harvesting there for biological supply companies, and apparently the lamprey stack up by the thousands underneath the falls, and you can just go pick them up with your hands. But as far as the nutrient input of lampreys, the only studies that I know of are the one study I referred to and then our back of the envelope calculations, but I'm hoping that when we have some better information, we can start to nail those numbers down better. But I think they're inconsequential compared to alewife.
There is a gentleman, I'm sorry he wasn't here tonight, he lives a couple of blocks from here, helps us out in Mayday, who used to teach science in the area, and he used to collect for biological supply. said he sewed bottle caps onto his gloves to better grip.
Oh, neat, yep.
It didn't sound like you were having a whole lot of trouble grabbing them there, or it didn't look like.
That's a way to do it, yeah, interesting. Yeah, we just use cotton or wool gloves.
But he was doing that in Bodenham, actually, and in Cooper's Mills is a popular spot.
Yep.
for sheepskin. Filming, how'd you do get some great shots there?
Oh, just
I could
to-
watch them all day with you.
Yeah.
How were those taken?
Just a simple camera that worked underwater. I don't even recall what kind it is.
And were you just with someone lying there watching?
Yep.
Or
Just me, usually. That's how I would spend a lot of my summers. The grad students would be off doing the hard work, and I'd be just sitting there watching lamprey.
Great. Thank you very much. So-
Anybody else?
So, let's let you pick a raffle winner for this fine vest we have here.
All right. Okay, and the winner is... Well, I know I didn't put my name in there,
I put your name in
well,
there.
I don't need that. Let's go for, how about Richard Brown?
Hey,
You get it.
All right.
okay.
Well, thank you so much again, and
Well, good, I'm glad you showed up on a nice chilly night.
yeah, it's chilly in here too, isn't it?
Hopefully you enjoyed it.
Thank you. Thank you.
No, you're very welcome.
How many years